Okay, we are now also on YouTube. Okay, so I think we can probably start. So thanks everybody for joining us from different parts of the world, I see, which is nice. So this is actually now the third year that we are doing this virtual seminar on complexity series from the Center for Complexity in Biosystems. We have now two new young co-organizers, Marco Zanchi and Marianna Duru, who will take over more and more the organization. So today, as a legacy, I start to introduce also because there is Miguel, who is an old friend. So I wanted to have the pleasure to introduce him. And so the seminars will take place more or less once a month, with sometimes maybe more, depending on how many speakers we have. They will take place on Zoom always. We have a website that you probably know because you have registered, so you get emails. You don't need to register every time. So once you are in the database, we'll send you emails. So you are in our mailing list forever. No way to remove you. So it's very complicated. Anyway, I'm joking. So, but so you can check your email or the website already for the next seminars that are already organized. So this is organized by the Center for Complexity and Biosystem of the University of Milan, but we are, of course, open to the rest of the world because of this virtual format. So today, as I said, we have the pleasure to have Miguel Munoz from the University of Granada in Spain. He's professor of theoretical physics, expert in statistical mechanics of non-equilibrium systems. I worked on many different problems, originally on absorbing state phase transitions and self-organized criticality. We wrote a couple of papers together now more than 20 years ago on the relation between these two subjects, between absorbing state phase transitions and self-organized criticality. And now Miguel, in the latest year, has applied all these ideas coming from StatMech of non-equilibrium systems to phenomena in the brain. And for instance, avalanches in the brain and other issues. So, and he wrote a very nice review that I suggest to everybody, that is been published 1 or 2 years ago on Review of Modern Physics on criticality in biological systems, which I think is very inspiring and useful to have an overview of the field. So today he will tell us about his latest work on criticality, renormalization group, and the brain. So thank you for participating, and please go ahead.
Okay, so thank you, Stefano. Thank you, Caterina, and all the group for inviting me. As I said, to be here, but here is in my office. So everywhere with you, with this broad audience. Okay, I cannot go backwards with this. I don't know why. No, I think I have to stop sharing. No. Can you see my slide? Can you see my slides?
Yeah, we see neuron, but
yeah, but I cannot go.
So maybe stop sharing and start again.
Yeah, stop sharing and I start again. Where is this? Share again the whole thing. And now I come here like this. Okay, now should be okay, right?
Yes.
Except maybe this, we don't need this. Okay, so I'll be talking about the brain and some aspects of its activity. And no, it doesn't work again. I don't know. Oh, maybe with the mouse. Okay, now it's working. Sorry. Okay, so here we have this front page of this article in Neuron a few years ago, where they talk about spontaneous fluctuations in the activity of the brain. So if you don't know it, it's something a characteristic of the human brain and also of the brain of all mammals, that our brain is never resting. It's always something going on. There is always some background activity. Even you are extremely relaxed, if you are sleeping, whatever you're doing, if you're alive, there is activity going on. And this activity here called the brain's dark energy, because it's not completely clear what's it useful for, it's very expensive from an energetic point of view. It consumes up to 20% of our energy. And when you're doing something like getting very concentrated, focused, and speaking, even a seminar, the increase in this amount is very tiny. So the bulk of energy is going to maintain this activity, and this activity can be measured in very different ways. Nowadays, looking at individual neurons, or more traditionally from a larger scales with fMRI or EEG, MEG, all these techniques where you look at the macroscopics. So questions that guide my research is, what's the functionality of this spontaneous activity? So what's useful for, and can we understand it as the result of a physical process? So if we have information about the neurons and their connections, can we understand physically how this activity comes about? Okay, so my interest in the field came from avalanches, as Stefano already suggested. So this paper here was really a breakthrough in the field. It's 18 years old already, I can't believe it. So Dietmar Plens and his PhD student, John Becks, were the first to analyze the activity first in vitro, then in vivo in the monkey of neurons. And what they saw is that in between periods of activity, as you have seen here in these movies that Dietmar passed to me, you see this, so you have time series like this. If you record activity, and from time to time you have a peak, that's the visible thing. You put this point here, and these points of activity in different places are grouped in clusters. So you can do the statistics of these clusters and see, for example, what's the size, how many of these electrodes got active at the given time as a function of the size. And you see a very nice power law that also obeys finite size scaling, meaning that if you make the system larger, you see larger events. Also, the times are distributed as power laws. So these authors here were the first to suggest that this could resemble a critical process, a critical branching process, in which activity does not spread too fast or too slow, just in a marginal way. Okay, this is taken from my review that Saperi was Stefano was kind enough to mention. This is the simplest possible example you can think of critical behavior in a non-equilibrium system. So imagine you have this process in which you have some active sites, and this can propagate activity at some rate, lambda here, or can disappear the activity at some other rate. So as a function of these two rates, lambda mu, you have a phase diagram. You can have either a lot of activity or just that everything dies, so some quiescent or absorbing state. If you want to look for avalanches, here we have space. Imagine a one-dimensional space. We have time here. If you put a seed of activity here, it propagates just for a short time if you are in this phase. So if lambda is not large enough, you create some outburst of activity, but it finishes in a short time. On the other hand, if you're in the active phase, you put some seed of activity, and it propagates deterministically, almost deterministically through all the lattice, invades all the space and reverberates forever. So this could be the equivalent of an epileptic state, if you want. And in between, you have this marginal propagation, which is much nicer, in which you have this fractal-like structure with clusters of all sizes. And if you come exactly here or around here, you see this power loss of activity with the same exponents, the same slopes here in this plot as observed in the neural things, right? Actually, the values of the exponents change a bit. This is a recent paper just to illustrate that there is a lot of activity going on. This is from 2 years ago. And in here you see that there is some dispersion in the exponents, but they all seem nearby or related. In any case, these examples I showed to you were with at some scale of resolution, local field potentials, but nowadays you can do it at an individual neuron level with optogenetics and these very advanced nice techniques they have. And you see these avalanches almost, quite generically. I think I skipped this. So that would be the idea of criticality as a microscopic point of view. You can also look at criticality, critical dynamics from a macroscopic point of view. I will not be talking about this much, but just to give you a flavor, let me just show you this research program in which nowadays with our technologies, you can measure very well the map of connections between different regions in the brain. You have thousands of these small regions, and you can know how much or how less are they wired together, okay? Then you also look for activity with the functional fMRI, magnetoresonance imaging, and you can see which region is correlated with which at the given time window or in general, and you can create these maps, matrices of correlations and correlation changing across time. And this is the map of a structural connectivity, who is connected with whom. So the goal is to find out what's the dynamical function such that applied to the network of connections, you reproduce the static or dynamic map of correlations of functional structures, right? So we have worked quite a lot on this possible mapping, and believe me, that if you tune it to be critical or almost critical in some sense, maybe in a sense of a synchronization phase transition, you get the best possible mapping. But what's this criticality useful for? Okay, the main message I want to tell you is that with criticality, you generate scale invariant objects. This structure, these clusters are scale invariant. So you can connect from the microscopic to the macroscopic, all the hierarchy of scales. And that potential is very powerful because allows you to integrate together the behavior of individual neurons with cognitive processes at very large scales, right? But people in computational neuroscience and also in computer science in general has realized that criticality has a lot of possible advantages. So actually it optimizes the possibility of having large responses, large sensibility. You can have, from this point of view of information processing, optimal capabilities, huge dynamical ranges. I mean, a lot of computational virtues can be associated to this type of scales. But this is quite speculative. Well, you can implement this in computers, but relating this to the brain, it's still in a stage in which there is a lot of speculation. And we would like to put this together we'd like to put together these ideas with data, right? So we have these two reviews. The one is by Thierry Morin and Bialek, Our Biological System Poised at Criticality, where they explore these ideas. It's almost, it's already 10 years old. And then it's my own, in which I discussed similar things a few years ago. Okay, so as I work here, we would like to connect this speculation, these ideas, abstract ideas, with data, with actual data. So in the last years, I've been looking desperately for good data, or collaborators with good data to put at work these ideas. And 2 years and something ago, the group of Kenneth Harris, which is a very strong group in London, wrote this paper in Nature, which is absolutely remarkable. It's something amazing, I would say. What they do is the following. So they take the mouse, and they have developed with some experimentalists these techniques. It's called Neuropic Cells. Neuropic Cells proofs in which they can look at the same time the activity of more than 10,000 neurons. So for each of them, you have a time series with its activity. Okay, the only thing you know is exactly who is connected with who with these proofs, but you have a large connections of neurons in different. You can have that in different parts of the brain, and you can follow the activity, and you can really understand things. Okay, so what these guys discovered was really amazing. This paper here is about the visual cortex, and the mouse was watching some images that they were presented to it. And then they recorded this activity and look at the correlation, or if you want, the covariances between each pair of neurons. So they constructed the covariance matrix. Once you have this covariance matrix, you can do something very standard, which is diagonalize it to do principal component analysis, to see which are the main dimensions in this in this large space of 10,000 neurons, which are changing the most when you present some stimulus, okay? So they do this analysis, and here is the different dimensions ordered by rank. So this is the largest one with the most variability. This is the other one. And they observe that this is a power law with an exponent nearby one. And we know that we physicists like power laws because many times they are related to critical points and scaling invariance. We like that. And look what they write here in the abstract of the paper. They said the evoked population activity was high dimensional, so many neurons were excited, but correlations obeyed an unexpected power law. The nth principal component variance scale roughly as 1/n. And this scaling is not inherited from the power law spectrum in the natural images. So it's not just a one-to-one correspondence with the external world, it's something intrinsic. But then the most remarkable part is that they were able to prove mathematically that if you want to have a nice representation, let me summarize this. This is messy. So the summary is that if you want to represent a lot of information in a covariance matrix, in a correlation matrix, you need this property. You have the exponent. You need the exponent to be close to 1, as they prove mathematically. This is required for the representation here, the representation of neural code to be continuous and differentiable. Okay, so now you are lost. You don't understand what I mean, but this picture can help a lot. So imagine that this torus here is a very abstract. It's a sketch of the neural representation. So in the space of neural activities, you would like to have something like this, that this is the point is representing this tree, and this point is representing this tree. You would like the two of them to be nearby together. So if you change from this to this, you do not want to change completely the space of configuration of your neurons. The same thing for these Nemo fishes, right? So when you translate this into a very large dimensional, you have this manifold of the possible states, and you want it to be smooth, okay? These properties are reflected in being smooth. So if you impose that the representation is smooth, then you get this conclusion that the exponent has to be close to 1, okay? So if you put here in this first column, if you put very complicated images, then the spectrum is close to 1. But if you put things that are much, much, much simpler, you can have larger exponents. So it's like if you want to compress a lot of information, this exponent of the correlation matrix needs to be very close to 1. If you just have little information to put into your neural network, then you do not need this heavy tail, this power law. You can have something become very fast, even exponentially. You don't need to recruit so many modes. So this is interesting from the point of view of principal component analysis. I don't know how familiar you are with this. This is very, very common. streamlined in data analysis. But imagine you have this data here in this two-dimensional plot, and they mostly vary along this direction. So if you do the analysis of correlation, you will see that this is the principal component, and then you have a second one with also some importance. So in situations like this, with just a few principal components, with the dimensionality which is reduced, you explain 100% of the variance, almost all variance. But the situation we have here is that the eigenvalues decay very slowly as a power law. So you have a full tail here of dimensions which you cannot just neglect. If you come here to this plot where I'm putting the cumulative variance that you have put together, you need to put essentially all the dimensions to get to 100%. Okay, so it's very different. Is the problem of dimensionality reduction is much richer in these cases where you have a power law than in cases where you have just a few principal components, right? So if you want to have a robust and a nice representation of inputs in the neural networks, you have to keep a trade-off. So you need to keep as much information as possible that would represent a flat distribution of PCA components. So you keep them all, but you don't want to put much weight on the finest details. So you want the slope to be tilted. So if you impose these two things, you get that the best thing is to work with this exponent mu close to 1, speed larger than 1. So what happens in critical systems? This is just an example of the easy model. The easy model, you can have the ordered phase at very low temperatures, and there you just have one mode. You have disordered phase, you have a lot of modes, but this is very flat. But if you are right at the critical point, you have fluctuations, clusters of all the scales. And if you do the same analysis that we just did, you look the correlation matrix, you diagonalize it, which means going to Fourier space here, you get the power law with some exponent, okay? So the thing that resembles the most what you see in real data is this type of critical structure. Okay, so actually we have, there is evidence there that there is actual scaling variance in this activity of the brain, but we don't have any idea of what type of dynamical process is going on there, and how much depends, all this data depends on the input you are putting, the images, and how much it depends on the intrinsic spontaneous dynamics of neurons. And we don't know if this thing is shared by other regions of the brain. This is for the visual cortex. So it's universal or it's not. Okay, so what I am going to do from now on is a couple of theoretical analysis of this and other data to explore first if there is scaling variance in the sense of physics. Second, if this scaling variance has something to do with critical behavior. Okay. So to look for a scaling variance, the tool that we theoreticians have is the Renormalization Group. And here we have Leo Kavanagh with his idea for the Ising model or magnetic systems. And he says, okay, if we want to understand collective properties, larger scale properties, critical properties, you should be able to replace all these little spins by large spins and write down some effective interaction, effective distribution of their probabilities. And if you do that in the right way, you should be able to repeat the process and describe the macroscopic physics in terms of the microscopic physics following this procedure, which is called Renormalization Group. And here we have Bill Bialek, who wrote this paper also 2 years ago, in which they applied this idea of Renormalization Group to data. So it's a phenomenological renormalization group. They applied it to data in the mouse, also in the mouse. So here is the same mouse as before. And now they measure 1,000 neurons. It's not bad. They do it in the hippocampus, before it was visual cortex. And the mouse is here doing something. I mean, this is very complicated. I don't pretend us to understand all the details, but the guy is moving in a virtual trap, in a virtual maze. So it's moving right there up and down. And while it's moving and doing something, they record the activity of these 1,000 neurons. So here is a set of just 8 neurons. The real world is much more than that. And what they do is the same idea of the Kamenov's blocks for renormalization. So they take 2 neurons, they put them together into a super neuron, if you want, and so on. So here's the 1 and 2, 3, 4, 6, and so on. And then they iterate the process, and from these 2, they construct new neurons. The technical challenge was that you don't actually know which neuron is connected with whom. So what they did is a trick that they put together the 2 neurons that are most correlated. Okay, so you take the most correlated ones. So it's like changing the usual criterion of physical vicinity by a criterion of vicinity in the space of correlation. But I mean, sounds like very reasonable, right? And then I have a little bit of technicism here, but don't get scared. It's very easy. So this is the activity of one microscopic neuron. This is the activity of the one which is most correlated with it, and you put them together in a super neuron at second level of the hierarchy, and you normalize it somehow. It's not important now. Okay, then you iterate the process and see, you want to analyze what's the distribution or what are the features of these activities at larger scales. If you look at the distribution of these activities at larger scales, you can do something. This is for technical reasons, but you decompose the probability to be silenced, so nothing happening in this super neuron, and this is the probability of some activity different from 0. Okay, whatever. So if you look at the probability of being silenced, you see this very nice scaling here. These are empirical data. And here you see the cluster size. So you start with individual neurons and you group them. Here is individual neurons, then you group in couples, 4, 8, 16, 8, and so on. And you see this very nice scaling here. And this is the distribution of the probability of having some value, normalized value of the activity. And you see this fantastic scaling. So all the curves, all the distribution probabilities at different scales collapse into a single curve or converge to a single curve, which is non-Gaussian, okay? This is important in critical phenomena. We have that if the system is ordered, it's Gaussian. If it's disordered, it's Gaussian. But if it's critical, it's non-Gaussian. So non-Gaussianity is something that we like, right? If you look at the variance of this distribution, you see, and you plot it here, and you see that it also scales very nicely. It's quite amazing. If the variables were perfectly correlated, you should have an exponent here 2. If they were uncorrelated, random, then it should be central limit theorem. It should be a scaling exponent 1, but you get something in between. So you are not nor uncorrelated nor fully correlated, and you have a wonderful scaling, which is non-trivial, in between, okay? Also, you can study time-dependent things, but I skipped this. So you can actually measure a number of these critical exponents. This is for the probability of being silenced, this is for the dynamical thing, this is for the variance, whatever. They repeat the experiment for 3 different mouse in 3 different conditions, and they see that the exponents are quite robust. Not exactly universal, but kind of, given the numerical precision and errors, right? I think I skipped this one. It's a bit technical. Well, this, I mean, I go very quickly through this for those of you who are more keen of theory. They do it also a momentum space renormalization group. And it consists in measuring the correlations in the same way you do in the AC model. And now I skip it because I was mumbling and going to mumble just too much. So essentially, it's the same type of thing they measure with this renormalization group scheme, the same thing that I explained you before that was measured by the group of Kenneth Harris when they were talking about efficient representation, this same exponent. So again, you measure the covariance matrix, you diagonalize it, okay? And you do it at the different scale of the renormalization process. you rescale, and all the curves collapse very nicely, okay? So this is the fact that they collapse, and they have a part here, which is a power law. And this power law is 0.7, in the other mouse, 0.7383. Okay, so let's pay attention to this exponent, because this is the same one that I described before in the work of Harris. And I said it should take a value about, nearby 1, but always larger than 1 to ensure that you are representing information in a nice way. So here it seems kind of universal with the renormalization group, but it's not 1. Okay, it's below 1. So next question we face are, number 1, okay, this study of renormalization group by the group of Bialek is for the hippocampus. So is it universal for other brain regions? Or if you do the same thing for the visual cortex that was done before by Harris, you get the same things? And then we have this conundrum. How can this exponent that we found be compatible with the optimal input representation that had a different value? Here it's smaller than 1, and before it was larger than 1. And if there is this scale invariance, has anything to do with critical? Okay, so let's go step by step. The first one is, for us, our first challenge was to look for data in different good data, very good data, in different parts of the brain of the mouse, okay? And luckily, there are many, many very good data in the Allen Institute and also by the group of Harris and some other few places, in particular this Nature paper 2019. They have studied many different regions in the brain of the mouse, like these ones that I have here, primary motor cortex, thalamus, olfactory areas, whatever. Each one is located here in the mouse brain. And we repeat it with the data. In particular, for all of these regions, we have sets of data with something like 1,000 neurons. So we can repeat the renormalization group analysis. We have to do some tuning of the method for it to work here, but it works. So look here, different colors are for different regions. All the regions, and these are the curves that I plot before for the renormalization group. So this is the distribution of activity in the different areas, and you see that this kind of universal tail, which is non-Gaussian, the Gaussian would be this one here. Here in the inset, I have for just one area, the motor cortex, the scaling for different sizes of the renormalization group, and you see that it collapses very nicely. And also the variance that I showed before with the nice scaling. Now I have the same scaling, but for 20 areas in the brain, right? And it's remarkable universal. It's not exactly universal, but kind of, right? There are a little bit more dispersion here in the probability of being silenced. So there are some regions in which it's more likely to be silent than in others, but also with some dispersion the scaling is good. And this is for the dynamical time-dependent part that I didn't analyze so far. And this is the part of the exponent that I like the most. This one of the covariance matrix. Once you diagonalize it and study the principal components, the dimensions, you always see this decay with the power law. The power law is always very nice, and in all cases, it's smaller than 1, okay? So it seems like a violation of the principle of Ken Harris that this exponent should be larger than 1. So 0.9, 0.8, 0.9, it's kind of universal, kind of robust, but always below 1, okay? All the exponents are below. And just for comparison, these are the works of Bialek in the Hypocampus 0.7, 0.8, which are compatible with the type of values we have. So we have this problem. How does mu smaller than 1 in the hippocampus, in the visual cortex, in all regions, while the recent theoretical results predict that if you want to codify information in a meaningful way, you should have mu slightly larger than 1. Okay. So these are data from the visual cortex of Harris' group. And we took them and analyzed them with the renormalization group. The funny thing is that, as I showed you before, they had results for real world images shown to the mouse, which are these ones here, but they also have results for low dimensional inputs. They were showing, for example, stripes to the mouse, right? And for the two of them, you have different spectra. I told you this before. I told you when the spectra is very complex, the exponent is near to 1, very close to 1. And when the input is not that complex, you don't have that much information to store, then this mu can be larger, right? So we repeated our RG analysis and saw that indeed the renormalization gives us quite indication that both of them are kind of scale invariant, but the exponent is different indeed, okay? So it's like there is different fixed points of the renormalization group depending on the type of input you are presenting in the visual cortex, right? And here, I think I'm missing something I should have told you. Yeah. Okay, so the key point, I think I'm missing here a slide. The key point is that in order to obtain these results that I'm showing here and get their very nice conclusions about the representation of inputs, they had to do some cleaning of the data. So what's the cleaning of the data that the group of Kenneth Harris did? They invented something called cross-validated principal component analysis. And essentially, I'm sorry that I'm missing one slide, but the idea is very simple, I tell you in words. What they do is that they repeat the very same experiment twice. So they show this, they show the same images twice to the mouse. And then having these two time series for the same input, they compare them, they do some kind of principal component analysis and are able to discriminate which part of the response of the neurons comes from the input and which part is not coming from the input. So it's noise, right? So repeating different times the same experiment with the same mouse in the same conditions, they filter what's the activity coming from the input and what's the activity which is not coming from the input. And that's very nice, okay? So if you take the raw data without doing this analysis, we ask Kenneth Harris for the raw data, they pass us the raw data, and we analyze them with our renormalization group and saw that they are universal, the same type of values we had for other regions of the brain, okay? So when you look at the full activity without using this cleaning, you see this kind of universal exponent smaller than 1, okay? Using their technique to remove noise and isolate the actual input-driven activity, we did the opposite. We invented a technique to remove the input-driven activity and just leave the ongoing spontaneous activity of the neurons. And we got an even smaller exponent mu. So now you are very confused. I'm confused also with all these mu values. So this table is an attempt to summarize what I said. So here, as a function of the input complexity, the more complex the image, the smaller this value. The less complex, the larger this value. Okay, but now here in this dimension here in green is the level of noise. So if what you observe is just white noise, this mu should be 0. What you actually see in the ongoing noise activity is mu 1/2. When you put together, this is the raw data where you have input plus noise. You have this 0.7, which is intermediate, and this is the value which is universal across regions, okay? So what we are seeing here is that we have solved this puzzle. When you put all the information together, you see this type of scaling, right? And why this is not compatible with the results of Harris is because they are filtering out the ongoing activity, right? Okay, so we have seen a lot of scaling, and then we wonder whether this scaling has something to do with critical or not, because there are different mechanisms, there are different ways in nature and in the way we model nature, in which you can generate scale invariants without having to resort to a critical point, to a point in between different phases. Okay, so this is the question. It's a scale invariant, okay, because we have seen that the renormalization group tells us that there is scale invariance, but it's also some kind of criticality. Okay, and for this, we use a couple of recent theoretical approaches. Both of them, the two things I'm going to show you now, rely on computing this object. This object here is called the spike count covariance, or lag time integrated covariance. It's different from the standard covariance matrix that I discussed previously. So the standard covariance matrix I discussed previously would be this object here, this C. So it's the covariance between neuron I and neuron J. And usually, before, I mean, all along this talk, I measure this at the same time, okay? So you measure the correlation between two neurons at a given time. Now what you can do is measure this with some lag. So if this, I mean, you understand what this means. One at time t and the other at time t plus tau. And if you integrate this for all taus, you are looking like the whole correlation between two neurons regardless of the time. So it's a very robust measure of how strongly two neurons are connected. And this is related, that this equation shows you here, with the number of spikes that you count for two different neurons and how they are correlated. In any case, the only one thing I want to understand, to emphasize, is that this new covariance matrix I'm mentioning here is slightly different from the one I talked about before. So if you measure this type of spike count covariance, what you have is that you can have, let me show you here, you can have a dispersion of values across your network, okay? And in this paper, these people, I mean, it's a group of Moritz Elias, they use techniques of spin glass theory and disorder systems, very clever, mathematically very clever techniques, to using the information of this distribution probability, so how the entrances of this matrix, correlation covariant matrix, are distributed, they can estimate what's the largest eigenvalue of your matrix of connections. And you can then infer how far you are from one, and one is the edge of stability. So it's like the maximum Lyapunov exponent. When the Lyapunov exponent becomes one, the system becomes unstable. So if you want, this is this theoretical method is a way to infer the dynamical states from data, okay? From data you measure this covariant matrix, and from statistical properties of this covariance matrix, you can say how far it is from the edge of stability. And we measure it for all the brain regions we have in the mouse, I mean, 20 something here. And we saw that all of them are very close to criticality. None of them is at one, so none of them is truly critical, but they all are, let's say, about 4% at the edge of becoming unstable, if you want, at the edge of chaos, as some people call it. Also, interestingly, if you look here, the larger the sample size, for those regions where we have more neurons, the closer is the estimation to the value 1. So probably the ones which are deviating the most is just because we don't have good statistics, because we have a very limited sample. So the conclusion is that all of the regions are actually can be inferred to be very near the edge of stability. Moreover, and this is a recent preprint, we have already finished our work, or kind of finished, when this preprint by the group of Sompolinsky and Yuhu came about, and they did something more. They have the same setup as before. They measure, again, this same covariance matrix, but they were able to predict that if the dynamics is critical, if you're working at the edge of the instability, then the power spectrum So again, the same idea. You take this covariance matrix, you diagonalize it, you put the eigenvalues ordered, and then you see a decay. Okay, this decay, they show mathematically that has to be a power law, like in critical systems, of course. And the exponent has to be something close to 5/3, so 1.66, right? So we went to our data of the brain of the mouse, we analyzed all the different regions, and one by one, we did the same analysis, and in all of them, we got pretty neat power laws. I think that these are experimental data, right? And the power law is pretty nice. And in all cases, it's close to this 1.66. Look, for the visual cortex in particular, is 166. The other ones, there is some deviation. This is slightly larger for some region that I'm not going to discuss here. But our theoretical analysis of empirical data reveal again that this is working at the edge of instability. It's working very, very close to the edge of becoming unstable, in between the stable and the unstable phase. Okay, so summary again, not only we have found very nice evidence of quasi-universal scale invariance using the renormalization group, but also we have given evidence that the scale invariance seems to come from dynamical processes operating at the edge, at the very edge, or close to the edge of instability. So we also explore the possibility of looking for this in machine learning, in artificial intelligence. In particular, we have this question. Okay, as I showed you before, there is this very nice result, at least nice, I like very much, the fact that if you want to have a nice representation of the world of some inputs, then you have to construct a covariance matrix of your data, of your neurons that has a covariance exponent, this exponent of the power law close to 1. Okay? So we said, okay, what if we construct some machine learning, going to do some complex task, look at the neurons, look at their activity, and try to see if they obey this or not, to see if this, our paradigm of artificial intelligence is completely different of the way the brain is working. To do this, together with my PhD student, Guillermo Morales, we constructed a reservoir computing. We computed a neural network, an artificial neural network, which relies on the paradigm of echo-state, an echo-state network, the paradigm of reservoir computing. I don't know how familiar you are with this, but I'm gonna give you a very simple explanation. So what we have is this network with some connections that are placed in principle randomly. And there is an external layer where we are going to put some input, okay? This input can be changed in time, and it's gonna affect some of the neurons, maybe not all of them. Then this will reverberate, the activity will here reverberate, go from one to another side, do something very complicated. And then you have some readout. So from some of the neurons in your reservoir, you project to the output. So the funny thing of this type of architecture, this reservoir computing architecture, is that you do not train all the weights here, as you do usually in neural networks, but you just train these external links so that the output is what you want. So depending on the input, you let it run for some time and you want an output. So you train just the red ones. You do not need to change the state of the reservoir, and it works very nicely if you do it right? Okay, so these are details of how we did it. It looks very messy, but what we did is we took the same images that Kenneth Harris and his group were presenting to the mouse, okay? And we trained our network in such a way that it learned to recognize them. Okay, I skipped the details. The only detail I'll tell you is that in constructing this reservoir, we can change its spectral radius. So this controls its stability. and also another parameter that controls the strength of the strength of the input. We can regulate with an overall factor. So we have the following very nice result, I think. Well, here I'm presenting results for a simpler task, which is just this, the discriminating the numbers, the digits in the MNIST database, okay? So we trained our network to do this, and we took something like 500 neurons, or 1,000, I don't remember the last results, how large the network was. We trained it, and here you see in this plot here, that we achieve, even with relatively small networks, and 98%, 97, 98% of success in recognizing digits. So our neural network works nicely. Also, our goal was not to achieve 100%, was just to get something to see how it depends on the internal state of the reservoir. This is raw. This raw is controlling the internal state of the reservoir, okay? So, for example, where can I show you? The blue dots is the performance. So when you get far away from this central region, the performance is not that good. So what's this central region? Look, for example, at the Lyapunov exponent. Lyapunov exponent is the orange curve, and here is where it's the limit of instability. So the edge of chaos is around here, okay? If you are exactly here, the representation you have, I mean, you look at the covariance matrix of your units in the reservoir, you diagonalize it, you look at the power spectrum, and you have this mu, and it's 1. I was really shocked when I saw this. So we were not tuning this to get this mu equals 1, but when you tune your network to work at the edge of chaos, at the edge of instability, automatically we get that the internal representation of the inputs, has an exponent close to 1. Still, the best one is not at the edge of chaos. It's a little bit in the stable region, okay? It's like a couple of, it's 93% of the optimal, right? So you have to be slightly below, and here you get an exponent 1.1, which is the same thing you see in the experiment, or 1.2, and slightly stable dynamics. So I think this is very nice. I like it very much. We have found that our artificial intelligence, which is it's a toy model of artificial intelligence, but it's doing something very similar to what the actual neural networks are doing. It's achieving the best performance by working close to the edge of instability, but below, and spontaneously, automatically, we are achieving representations with this value, which is optimal, okay? So take-home messages. First, all of this is analyzing data and looking at data with tools from theoretical physicists, from theoretical physics. We found that there is very good scaling, and that you can study it with a Renormalization Group, and that the scaling is quite universal. So you can look at different parts of the brain of the mouse, and you see very similar critical exponents. It's not like in physics that you get the critical exponent with 5 digits or 3 digits, and it's always that. Here we have some dispersion, but the dispersion is always, let's say, below 10%. And we have a lot of numerical errors, and the system size is about 1,000. So given these constraints, the results are supporting universal scaling. Second, it's not just that it's scale invariant, but we found evidence that it's working at the edge of instability. So it seems that the brain actually is at the edge of changing regime from some stable to some unstable. And finally, we constructed our toy artificial intelligence model neural network, and we find that the best performance is obtained in a very similar regime, in which we are close to the edge of instability, and we have this property of the optimal representations. And I think that's it. I just have to thank overall Guille, who has done an amazing work in doing all these analysis, collecting data, doing DRG and all the techniques. And also my long-term collaborator, Serena, who's now in Columbia, in New York, becoming a real neuroscientist, who also helped us a great deal. And that's it. I can take some questions if you want. Thank you.
Thank you very much. It was a very nice talk. I have some questions, but let me first ask if somebody else has a question. Guido raise his hand.
Thank you. So my question is whether it is known if the dimensionality and the topology of the contact network underlying the signals you studied, how it looks like, if it's more like a beta lattice or it's loopy or whatever.
I mean, the actual neural networks.
Yep.
Oh, they are extremely recurrent. So it's a small world, everything connected to everything on a local scale.
So that's the same in your artificial neural network?
No, it's not. No. We have long-range corrections. So it's, I mean, it's the same thing as in experiments. We don't have something like a beta lattice or something like that. No. Everything is interconnected. Okay. Actually, in some of the studies, we connect all to all. But I think in that case, the efficiency is worse because then it's a bit of a mess and the convergence is slower. So you have put that sparse network, but essentially it's an Erdos-Renyi network. I don't know if I answered your.
Yeah, thank you.
Thank you. Other questions? So I have a few questions while the other think. So first question is, so you show that there is a difference between what you call noise, which of course is not white noise, yes, and a response to an input. Okay. So do you have any idea of why there is this difference and what does it reflect? Because of course this noise is not a noise. I mean, is not a background noise. It's something that is somehow since it's very correlated and so on. So,
right.
And the other noise is response to input and has different statistics, or if you want. So how do you understand this difference and how you picture that?
Okay, I would love to understand this difference. So essentially what we see again is that everywhere in the brain you see this exponent, okay? And if you filter to detect where is the information represented, you are able to see this nice mathematical feature. But what you actually see everywhere is this. And here we have a mixture of the inputs and of the ongoing activity. My guess is that in every region of the brain that you are looking at, you are seeing a lot of inputs from many other different regions. So even in the visual cortex, it's known that you have inputs from whatever other region because you have to integrate activity. And I mean, the different regions are not isolated. They are all connected. So you have many inputs together. And the effect of all other inputs, even movement, for example, is translated into, I mean, if you move, essentially every part of the brain is going to get information about the fact that you're moving, just to control vision, for example, or to control audition or whatever. So the combined effect of all these many complex different inputs from different sources and of different scales somehow gives rise to this everywhere. Now, if you were able to switch off all inputs from the external world and you were in a black box, then everything would be intrinsic. But intrinsic for the whole brain does not mean intrinsic for a given area because it's exposed to the other areas. So final answer to your question is, I don't know. It's a mess. You see that this is kind of universal, but it's not clear if it's just a spontaneous local or spontaneous combined. I don't really know.
Okay, Michele Colombo has a question.
Thank you very much. I was actually going to ask something very close to what you asked now. So this was the interesting summary picture that made me thinking. So yeah, you were saying that basically the spontaneous activity, when filtered out from the inputs, would just have this mu near 0.5, and when instead you would only consider the reproducible part due to the visual input, then you would have this moves just slightly above 1. So that it seems like the external part is that it's driving the brain, if you want, is the part that actually follows the prediction of having this exponent above 1, right?
Yeah, exactly. So it's just when you filter out noise that you see this smooth representation. But the smooth representation consists of noise itself because it's just the speaking of neurons. But there is a regular part that is the one codified in here. And then there is, on top of that, there is noise from other regions or from just noise, I don't know. And when you put together, you see this.
I see. So since you're an expert in renormalization, then I was going to ask something about a different scale of brain activity. So when we look at the electroencephalogram and we look at a different exponent, just the Fourier spectrum has another kind of power law, and it can be argued whether it's a true power law or not, but the region that we're sure has an exponent that in the waking state is between 1 and 1.5, and then certainly when it goes above 2, and then the person is either fallen asleep or has taken some anesthetic drug and so on. So I was going to ask if you know what would be the theoretical prediction for the exponent there, if it was also related to this 5/3, or if there's a critical exponent in this with respect to this Fourier space.
Yeah, I'd love to know the answer to that. So actually, you're talking about what people call 1/f noise, right?
Yeah.
So there has been renovated interest in that part of the spectrum, because at some point people were just focusing on the peaks, and now I've seen a lot of activity of people trying to figure out what information can you get from this power law. But the question there is that there are so many mechanisms that has been described to get power laws there, because in the EEG you are looking at the extracranial scale, and there are filters, for example, coming from the skull and from the other parts. And it seems, I don't know the details, but it seems that these filters can induce the power law. So it's very messy. I spent some time and I put one of my students to dig into the literature. And after a week or two, we said, this is just too messy. It's difficult to get a clear idea of what this exponent is coming from. That does not mean that there is no something very interesting there, but I cannot answer your question. I'd love to, but I can't.
Thanks. Marianna has a question.
Yes. Hello. Thank you for this presentation. This area is new for me, so I want to ask you something more biologically. Have you correlated this data, or the authors who performed the experiments with the mouse, with the age of the mouse?
With the age, no, not really. In the Allen Institute, they have plenty of data, but we were just looking for collections in which you have a lot of neurons, and those collections are limited. So we were lucky to find representatives of all these regions, but now if you want also the dependence with the age, I think we have to wait for the experimentalists to have more data. But it would be very nice, also because they say that some people argue that the better performance in the jout, let's say, it's achieved when you're closer to criticality, and as the generation goes on with age, the dynamics gets farther and farther away from the critical state. It would be very nice to verify, but I don't think we have the data yet.
Okay, thank you.
Marco?
Yeah, I have a more technical doubt. So I didn't understand why. What are the advantages of using lagged correlation metrics?
Advantages. Okay, when I started reading these papers of neuroscientists, I was very confused, because every time they measure a a covariance, they measure something different. And we physicists are used to measure standard correlations, like I at time t and xj at time t, and that's it. And you measure a time-dependent correlation, and if you want the stationary state, you measure that. Okay, and that's the one that I have used in the talk for BLX and for the RG, Renormalization Group, for the Kenneth Harris. All that is with the standard measurement of the correlation function. But if you go at the papers in neuroscience, many times what they count is the number of spikes of a given neuron in a given time interval. And they correlate this thing. And this thing gives you a slightly different idea of the type of correlation. So one thing is instantaneous correlation, which is the one we use in physics usually, or with some delay, but it's a fixed time correlation. But if you integrate across time, it's like a little bit, you put together all the possible specific time correlations between the two neurons. So you integrate all possible correlations and you get like a supra correlation between the two neurons. So independently on the time delay, how much are these two neurons correlated? And with this, you can compare how much are these two neurons correlated, how much are these other two neurons correlated. So it's like the advantage, to answer your question, is that it allows you to discriminate neurons as a function of pairs of neurons as a function of the strength of the correlation. I don't know if I make sense.
Yeah. Thank you very much.
Okay, actually in neuroscience, they speak quite generically of noise correlations and input correlation or signal correlations. One is the one physicists like is signal correlations, and the one that I just discovered is noise correlation. So this terminology, but they use these two things. Mm-hmm.
Thank you.
Other questions? So I have another question. So this strong universality, in your opinion, comes from the fact that in the end it's all mean field in some sense, in the sense that everything is connected with everybody. So, well, it doesn't matter whether it's the cerebellum or the hippocampus. It's always a bunch of neurons connected all together.
I don't know. I would like to understand better this.
So exactly, but this is just finite size scaling, or it's something intrinsic. So
okay, we don't really know. We don't really know. It seems, I mean, if you look at different colors here are different regions, and depends. If it's just finite size scaling, essentially they will all go to one. But if you have beta data, you could discriminate between them. For example, here for the same size, this one has a smaller exponent, has a larger distance to criticality than this one, and the sizes are comparable, right? So maybe we could understand something from the dynamics. Let's say that the motor, I don't remember which one is this. I mean, we would like to explore what's the connection between the different regions. For example, if sensory regions could be closer to criticality than motor regions or something like that. We are trying to explore that, but it's not easy. It's very messy. So far I don't have a good answer. Probably there is some kind of central limit theorem of fluctuations in interconnected systems that led you to this super universality. But I don't know. I don't know.
So, but always a connection with criticality, you know? So you say you're far from the critical point, no? But this is something that typically is non-universal, no? The fact that you are...
yeah, how far you are is non-universal, exactly. But maybe you could...
What is universal is maybe the exponent at the end, when you are a critical point. But here you're arguing a different things. You're saying how far I am from the critical point. So maybe different regions are different.
Exactly. Maybe in some regions you could need more stability. Imagine that here you don't want, I don't know, if you have the clock of your brain, no? the what's called the suprachiasmatic nucleus, where you have your internal clock. Maybe there you don't want criticality. You want something that goes with a very fixed scale, no? So that would be nice to understand. If different regions with different purposes can be more far away or closer to critical level. but all of them are nearby. Okay.
Yeah.
But now what this nearby means, of course, could be quantified. And discriminating between regions as a function of distance to criticality would be a dream.
And another question, more technical. So you mentioned that at some point you mentioned an Ising model, and that it's the same thing, and you have 2 minus eta as an exponent.
Yeah.
But in that case, if I understand correctly, you went very fast, but a correlation function in Fourier space, no?
Yes.
Is it the same thing as the rank order PCA?
Yes, it is.
It is.
This is a slide that I skipped. This correlation function in a system which is translation invariant. If your system in physics is translation invariant, it just depends on the distance, right? Okay. And if you do the Fourier transform, or if you want, this is a matrix that depends on index I and j, you diagonalize, and the diagonal part is the Fourier.
Yeah, but K is not the rank, but it is the rank because the
K it's related to the rank. Here is the K and here is the rank, but the rank goes with the size of the shell in K space. I mean, it's proportional. There is a close relationship.
Okay, thank you.
Right?
Just to understand.
Yeah. Yeah, let's say for a fixed modulus of K, you have a fixed number of eigenvalues, and then you can order them as a function of the rank. So essentially it's the same thing.
So basically you are measuring eta in all this brain.
Yeah, exactly. I'm measuring eta.
Okay. So mean field would be
mean field would be eta 0.
So
exponent 2. Yeah. Actually, I have the relation with mu somewhere. I mean, between mu and eta in a hidden transparency.
Remember our very cited paper where we give all the scaling relations within the different exponents. Yeah. Everybody likes it because nobody knows how one exponent is related to the other one.
Using that paper of us, I wrote down the relation between this mu and this eta. Yeah.
Okay. So other questions? So I also remind you that the talk is in YouTube and will be available on YouTube. Okay. So if you miss some details, you can always go back and check it. And